So the title of my talk is Solvation Thermodynamics of Oligoglycine, Implications for Aggregation and Collapse. And what we're going to be talking about today is the thermodynamics underlying the process in which oligoglycine is transferred from the gas phase to the fully solvated state. Now before I get into this, I'm going to talk a little bit about the biology that's motivating my research, and this has to do with disorder in proteins. So intrinsically disordered regions, or IDRs, in a protein are characterized as highly dynamic and flexible. And the extent of disorder can range from no disorder at all, that is the nicely well-structured protein, to entirely disordered, with all flavors in between. Now, IDRs play an important role in facilitating molecular signaling networks. They often directly bind and recognize DNA or protein targets. They're also the site of allosteric regulations in these networks. And it's not surprising then that they've been implicated in a number of diseases. So if we're going to be someday as successful in targeting IDRs with drugs as we are in targeting well-structured proteins, we need to have an extensive understanding of the thermodynamic and structural properties of IDRs. In 2002, Uversky proposed the protein quartet hypothesis. And essentially, this attempts to characterize the structural states that a protein can assume. With on top, we have the well-structured or ordered protein and three disordered states on the bottom, all differing in their extent of collapse or compaction. The molten globule is highly compact, the random coil is more extended and flexible, and the pre-molten globule falls somewhere in between. Now, the occupancy in and transition between these states depends in part on chain length and sequence composition, among other things. And we want to understand, and each of these states gives rise to a particular unique function. We want to understand what drives proteins into structural states and what the role the solvent plays. And the main question we're interested in asking is whether the thermodynamic mechanisms that drive the proper folding of a protein into its native state are similar to the mechanisms that drive an IDR to its native collapse state, as well as IDR-mediated aggregation of proteins in a pathological state. We know that the hydrophobic effect, or a negative entropic penalty paid upon solvating a protein, is a major player in driving the proper folding of a protein, but we don't have as good an understanding of how important these effects are in these two mechanisms over here. And so to start to elucidate some of these mechanisms and better understand them, we're going to use oligoglycine as a model. This is a polypeptide composed of only glycine amino acids. We're going to be looking at various different lengths of oligoglycine. This is the protein backbone. It doesn't have a bulky, complicated side group. IDRs are enriched in glycine, as well as oligoglycine tracts. The longest oligoglycine tract I've seen in a protein sequence is Gly25. And a high glycine content has also been associated with more compact IDRs. So experimentally, what do we know about oligoglycine? We know that as chain length increases, oligoglycine becomes highly insoluble at finite concentrations. And at around Gly5, they start to aggregate and form these large aggregates. We also know from single molecule experiments in highly dilute solutions that oligoglycines will collapse. So what's causing these things? What's the role of this entropic term in driving these processes? And what we want to get at is the solvation thermodynamics, both of its enthalpic and entropic components as a function of chain length and other things. And so to do this, we're taking a computational approach. We define our reference state as oligoglycine in the gas phase, not interacting with our box of water that we're going to be simulating. And then the final state, we have oligoglycine fully interacting with all the solvent molecules. And this interaction energy can be modeled as the sum of two terms, a van der Waals potential energy and electrostatic potential energy. In the reference state, again, they're not interacting, so the interaction energy is zero. And we're going to computationally connect these two states through a non-physical pathway by means of two coupling parameters, lambda van der Waals and lambda electrostatic. When both of these are zero, we're in the reference state. When both of them are 1, we're in the final state. And we're going to gradually increase these between 0 and 1, perform a simulation, a molecular dynamic simulation at each intermediate lambda, calculate the average of the derivative of the potential energy with respect to lambda. It's not terribly important to notice, but we integrate over all of the intermediate lambdas, and that'll give us our van der Waals solvation free energy and our electrostatic solvation free energy. So to give you a better idea of what this looks like, I'm graphically depicting it. In the first pathway, we set lambda electrostatic to 0, and we gradually increase the van der Waals lambda between 0 and 1. Again, at each of these intermediate states, we're performing an MD simulation. Plug into the equation I showed you on the previous slide, integrate, and that gives us our van der Waals solvation free energy. In the second pathway, we set lambda van der Waals equal to 1, and then we gradually increase lambda electrostatic between 0 and 1. Again, plugging into the equations, we get the electrostatic solvation free energy. So these are our two pathways. This is how we get the two components here. They sum to give us the overall solvation free energy. And for the sake of time and the remainder of the talk, I'll be focusing only on this solvation free energy term. So to outline exactly what we did, we calculated the solvation free energy for glide 2, 3, and 4 held in a rigid extended conformation. Often these positional constraints are imposed when doing some of these free energy calculations. But we want to understand how flexibility or disorderedness affects these calculations. So during our MD simulations, we allow glide 2 through 5 to be completely flexible. That is, there's no positional constraints imposed. We perform these simulations with two different force fields, CHARMM36 and Amber FF12SB. We want to be able to control for any force field effects and compare these. We do the simulations at 300 Kelvin and 320 Kelvin, two different temperatures, and we calculate the solvation free energy. And we perform a thermodynamic decomposition of delta G into its enthalpic and entropic components. And we use two independent methods to do this. The first I'll be referring to is the finite difference approach. Here we're calculating the solvation entropy as the finite derivative of delta G with respect to T. In doing some arithmetic, we get the solvation enthalpy. And we're estimating this at 310 Kelvin because the finite difference of the numerical derivative is more accurate at the midpoint of 310, right in between these two temperatures, and approximating delta G at 310 as the average of these two. In the second approach, I'll be referring to as the endpoint energy approach, we actually directly calculate the solvation enthalpy at the two temperatures, simply as the average of the total potential energy in the final solvated state minus that of the reference or gas phase. Again, arithmetic will give us delta S, or the solvation entropy, at 300 and 320 Kelvin. And this is where blue waters really came into play. Our system sizes are small. I mean, we can get around 100 nanoseconds a day pretty easily, but the problem is these intermediate lambdas. And in total, we had to do 1,784 simulations for an aggregate time of 92 microseconds. So blue waters, its high throughput and ability to run a large number of jobs concurrently really made this project tractable or feasible. So now jumping into the results, we're going to be looking at figures like this, so I'm going to just briefly walk you through it. On the x-axis, this is the number of residues in our polypeptide, and the number of glycine residues. The y-axis is energy in kcals per mole. On the left are results for CHARMM36. On the right are results for FF12SB. Green is the solvation enthalpy. Blue is the solvation free energy. And orange is minus T delta S. So the top, this one and this one, add to give you the solvation free energy. The solid lines are for the results for the rigid extended oligoglycine, and the dashed lines are for the flexible model. So briefly now, let's talk a little bit about the results. We see that the solvation free energy is negative and favorable, and it continues to decrease as the number of residues increases. So presumably, the solubility of oligoglycine should also improve with chain length. We see that the scaling of delta G is primarily enthalpically driven, and only a moderate compensation for by this negative entropic term. And both force fields are actually capturing these thermodynamic scaling profiles extremely well, extremely similarly. Now, going on to the endpoint energy approach, results for 300 Kelvin are on top and 320 are on the bottom. Essentially, we're seeing the exact same thing. Actually, I forgot to highlight. that when we compare the rigid to the flexible results, we see that the scaling of the solvation free energy is quite small. However, flexibility is really affecting the enthalpic and entropic components to a much greater extent. But due to their compensatory relationship, they actually add and result in much smaller differences in the solvation free energy. So coming back to the endpoint energy approach, we see that these same trends hold. And what's particularly attractive about this approach is that it requires half the number of simulations to get at the solvation enthalpy and entropy as the finite difference approach. And in fact, even when we look at the individual components, we see that flexibility is even to a greater extent affecting the scaling relationship. So now let's think about or evaluate our results in the context of the observed insolubility or aggregation of Gly-5. That's observed experimentally. So to remind you, our results are at infinite dilution. We're doing these in simulations. And we know that the solvation free energy of Gly-5 is favorable. And we know that this is primarily due to the ability of Gly-5 to form favorable interactions with the solvent, and this offsets the entropic penalty. So if it's not this entropic penalty or a hydrophobic effect that's driving the collapse of Gly-5 at finite concentrations, then what could it be? One reasonable explanation is that peptide-peptide interactions are actually outcompeting the favorable peptide-solvent interactions. And this is done in a concentration-dependent manner. As concentration increases, the number of Gly-5s increase, as well as the potential interpeptide interactions. And these are actually favored or outcompete the still favorable peptide-solvent interactions. Now we can do the same thing for the observed collapse of long oligoglycines in highly dilute solutions. And we can think about this in two different cases. One is our scaling relationships hold and can be extended out to longer chain lengths. And we could similarly make a similar argument as I did on the previous slide that peptide-peptide interactions are outcompeting peptide-solvent, but in a chain length dependent manner. So as chain length increases, the number of intrapeptide interactions increases, and these outcompete the interactions with the solvent. Now, in the second case, we can imagine that these scaling relationships then don't hold, that at some point the entropic term takes over, and that collapse is an entropically driven process. This would kind of be a bummer because then we couldn't be able to predict some of the solvation thermodynamics of longer ones that are much more difficult to access even through simulation. So now if we compare, let's compare the two different force fields. Now I'm showing things a little bit differently here. I'm plotting the results for the flexible oligoglycine models and color coding according to force field with CHARMM36 in blue and Amber FF12SB in orange. And basically what we see is that they are remarkably similar, especially the solvation free energy. Some minor differences in the two components. Now what's interesting about this is that in a previous study, we highlighted the force field dependent properties, structural properties of Gly-10 using molecular dynamics simulations. Here I'm showing 5 representative structures from a clustering analysis from a microsecond simulation with CHARMM36. And we see that CHARMM36 is sampling much more extended solvent-exposed structures than Amber FF12SB, which is sampling a greater population of more collapsed compact structures. And even when we get down to as small as Gly-5, we see the structural properties are also starting, the differences in the structural properties emerge as well. This is a histogram of the solvent-accessible surface area taken from the trajectories. We see that Amber in orange here is sampling much more compact structures in which the peptide is buried or is excluded from the solvent. And so this might be indicative of a degenerate structural ensemble to thermodynamic relationship. That is, these force fields are parameterized using different methodologies, targeting different sets of data, and generate different ensembles. Yet we're seeing extremely similar thermodynamic profiles. So this would complicate trying to connect structural mechanisms to thermodynamic mechanisms. But I think clearly this warrants some more investigation, especially out towards the longer chain length. But even if we attempt to, by eye, extrapolate these, the very minor differences would still be considerably similar towards longer chain lengths. And so with that, that about wraps it up. I'd like to thank my PI, Dr. Monty Pettit, my lab, and all the members who have helped me with these projects, and importantly, the Blue Waters Graduate Fellowship for their support this last year, and the allocation really made this project possible. And it's been an honor to be able to fund my own research this past year. And with that, I'd be happy to take any questions.
Great talk, by the way. So I know you said you weren't going to talk about the decomposing the calculations into the electrostatic and van der Waals components, but maybe you could say just a thing or two about how that plays into the differences in maybe the force fields.
Sure. It's like I planted you in the audience. I have these supplemental slides here. So what we find is here I'm showing the van der Waals solvation free energy on top, the electrostatic in the middle, and then the overall. The electrostatic term really dominates the scaling of the overall solvation free energy. If you compare these two scales, I mean, this is pretty negligible. This happens to be where we see some of the major differences between the force fields. Granted, they're on the scales of 0.5 to 1 kcals per mole, but the electrostatics really drowns it out. Now, for the solvation entropy, it's kind of a different story. The van der Waals term is contributing at least twice as much to the overall solvation free energy, and it's really dictating how the overall solvation entropy scales with chain length, and as well as contributing to the separation between the rigid and flexible models. And then flip that, because it's a compensatory relationship, the solvation enthalpy is dominated more by the electrostatics than the van der Waals.
You mentioned the Blue Waters with some characteristics that were instrumental in getting your problem solved. One of them was concurrent tasks. There was another one too. Or actually, you said concurrent jobs. Were they jobs or tasks, or did you run mostly within a larger job, or maybe just go into more detail there?
Sure. So since these system sizes were pretty small, it didn't require that many nodes. I think I was running at most 5 nodes. But I was able to get, I don't think I hit the actual limit on the number of jobs I can be running, but at least 50 at a time, between 50 and 100 simulations at a time. So that's where that parallelization came into play, was because I can run at each of those intermediate lambdas, they're independent of each other, that I can actually get all those done in parallel rather than having to wait for one to finish and then, or not even be able to run that many jobs. It would really slow down everything. So, things like the queue wait time, that really, I was able to get this thing churned out pretty quickly.